Welcome back. I am thrilled to introduce our next presenter, Mark Musen from the University of Stanford. He will be talking a little bit more about ensuring the quality of scientific data, a very important component to what data-centric principles are about. So Mark, it's all yours.
Thanks so much for that introduction, and it's really wonderful to be here. I'm giving you an opportunity to hear more about an application that has as its center the idea of maintaining data quality, but one that does so in a data-centric way. And so I'll be talking about a data-centric architecture for maintaining the quality of scientific data, something which actually is really important for all of us as we rely increasingly on science for everything around us in the world. The buzzword that has permeated discussions of scientific data is the question of whether data are fair, the question of whether data are findable, accessible, interoperable, and reusable. And over the past 5 years, there's been this incredible hype about the importance of making data accessible so that those data can be re-explored by other investigators, so that the experiments can be verified, and so that we can have more confidence in the kinds of results that scientists get when they do their work. The idea of having data that are fair is something that has basically been adopted by almost everyone. Everyone claims that his or her data are more fair than anyone else's, and has led to the enumeration of what we call the FAIR guiding principles. So the next slide, where the people who first defined the idea of fairness articulated a whole set of ideas to make data fair. The idea that data and metadata should be assigned globally unique and persistent identifiers, that the data are described with rich metadata, that the metadata should be clearly and explicitly include the identifier of the data they describe, and so on. When you look at the FAIR guiding principles, we're not going to go through all of them. What you see in the next slide is that almost all of those FAIR guiding principles have to deal with metadata. And metadata, the data that describe data sets, turn out to be the essential component of what make data fair, what make it possible for other investigators to understand what scientists have done, and to make it possible to be able to reuse data to make new discoveries. What that means is that we care a lot about how data are described. And when we look in the next slide on a real example of the metadata that accompany data from an NIH database, what we see here looks pretty normal. We can see that we have a set of attribute value pairs. We can see that there's a cell line with an identifier. There's a culture collection with another kind of identifier. This particular repository where these data are stored at a company that will remain nameless. There's a kind of tissue that was biopsied and so on. If we go to the next slide, what we see though is that what looks pretty good to us at face value actually has a whole bunch of errors in it. The disease is listed as carcinoma hepatocellular. Usually we don't put the adjective after the noun, and that would, if someone were searching for hepatocellular carcinoma, that term would never come up. The ethnicity is given as Japanese, which is certainly a nationality, but probably not what most of us would call an ethnicity. The age is given as 57 year, not 57, not 57 years. And again, if you were to search for this kind of stuff, you wouldn't find it. There's a real challenge that we face, because what happens when scientists are creating their data sets, they end up generating metadata that we would say is really terrible. They view their primary work as publishing papers, not leaving a legacy of reusable, clear, and accessible data. The sponsors or the management in the private sector who require data sharing are really enthusiastic about the idea of fairness. But often, and actually more generally, they don't encourage the use of research funds to actually pay for this kind of work. Creating metadata themselves is really hard, and I think that's probably the fundamental issue here. In fact, at least in the biological world where I work, when people are submitting data to repositories, usually they're filling out spreadsheets. And they're filling out spreadsheets in ways which really makes it really hard to know what goes in each cell, to be confident about how things have been filled in. And more important, makes it really hard to know what controlled terms one might use to define the metadata. And without controlled terms, as we alluded earlier, doing the kinds of searches that are necessary to find existing data sets is really tough. What that means is that if we do analyses of existing data sets, we find things are actually quite problematic. The Gene Expression Omnibus is a online database of functional genomic studies. And if we look at the way that the age of the subject for the data sets in GEO are described, what we see are horrible things. Well, not horrible, but we see enormous variety. We see age and age and age in all caps, and pop age and age after birth and age in years. And again, what do we search for if we're looking for the age of a patient being in a certain range? What do we do if we're really concerned that we want to get all the possible data, and yet we know that they're going to have all these different variations, and we know that there can be lots of typos as well. We, in fact, did a very careful analysis of an NIH database known as Biosample, and we see that they're absolutely dreadful. That 73% of the Boolean metadata values are not actually true or false. They have values like non-smokers, former smoker. 26% of the integers are not really even numbers. They're things like JM52 or PIG. 68% of the metadata entries that are supposed to represent terms from biomedical ontologies don't actually do so. They're text strings that seem reasonable when the scientists are creating their metadata, but actually are not searchable, aren't conformant with standards, and really make it such that if people want to get access to data in a reasonable way, it's really, really hard. What that means for us is that if we're looking for data that are findable, accessible, interoperable, and reusable, we have a real challenge. We have to think about what are the requirements that are necessary in order to allow science to get access to those data. They need data that are obviously represented using standard ontologies that describe what exists in a data set completely and consistently. And at Stanford, we've been working for many years on trying to enhance that situation by collecting the world's biomedical ontologies. Fortunately, in biomedicine, we're in a good situation because we have lots of biomedical ontologies that are available to us. 300 years ago, Linnaeus started to work on his ontology of speciation. And now in biology, everybody knows about species and genus and family and order, and can know how to refer to animals and other life forms precisely using Linnaeus's taxonomy. Bioportal is our repository of all the world's biomedical ontologies. And if you go to bioportal.bioontology.org, you'll get access to more than 900 ontologies that are used widely in biomedicine, and be able to access these in a very straightforward way using a common visual user interface and also a common API. What we see, for example, is the Bioportal gives you access to current procedural terminology, which the AMA uses to describe medical procedures, MEDRA, which is used to identify adverse events when information gets reported to the FDA, and lots of other ontologies, including SNOMED clinical terms, probably the most commonly used ontology for describing the details of clinical cases, all of which are obviously essential for making our metadata fair. Second requirement is to describe properties of experiments completely and consistently. What that means is not only do we want to have ontologies that give us standardized terms for use in our metadata, but those metadata themselves have to have a form that is standardized and one that we can expect to allow us to search for metadata and to integrate them. So if, for example, we think about gene microarrays, which have been around for about 20 years now. They represent enormous high throughput experiments where we're able to see how genes get turned on and turned off over the course of some intervention. The gene microarray community got together and said, look, we're putting all these data online, but we need to understand what the data are. We need to understand what's the substrate of the experiment, what array platform was used, what were the experimental conditions. Basically, what do I need to know in order to replicate this experiment? And so what the microarray people did is to say, look, we can't just put our data online. We have to have metadata that conform to a very well-defined structure. They created something called the Minimal Information About a Microarray Experiment, or MIAMI. And MIAMI basically lists all the things that ought to be in metadata that would allow somebody else to figure out what experiment was actually performed. But it didn't start an end with Miami. This became a trend in all of biomedicine. So now we have minimal information about T cell assays or MIATA, minimal information required in the annotation of biochemical models or Miriam, minimal metagenome sequence analysis standard or MiniMES, and all kinds of these things. And what that means is that the scientists who are involved in making their data available recognize the importance of creating structured reporting guidelines so that people can make sense out of their data. The problem, of course, is that these minimal information guidelines are not really computational models. They're just checklists of the kinds of things that investigators need to include in their metadata. They don't give us any kind of detailed information about the metadata elements that we expect. They don't provide data types for valid metadata entries. They just say what the entry should be. And obviously it takes a lot of work to convert such a checklist into something that's computable, which is obviously something that we're very concerned about in our work at Stanford. So to make sense of metadata, we need computer-based standards. We need to have standards for what to say about experimental data. We need a language that allows us to say those things in a standardized way that is machine interpretable. And we need to be able to structure metadata in a way which is familiar and constant across different kinds of experiments. And we obviously need tools that make it easy to apply these standards, and we need a scientific culture that values making experimental data accessible to others. That cultural shift is really critical, not something that we can solve easily with technology, but the other elements we certainly can. At Stanford, we've been working for the past 6 to 8 years on a system called Cedar. And Cedar has the requirement of making it palatable to describe those experiments completely and consistently. And that's really where we need good tools. And so Cedar, which stands for the Center for Expanded Data Annotation and Retrieval, represents a project that's been funded by the NIH with the goal of making data fair. And in the Cedar approach, we basically can look at things in terms of 3 different steps. In the first step on the left, we have a pane where we show the authoring of metadata templates. We basically are able to take reporting guidelines or minimal information models like Miami and MiniMES and Miss Fishy, and we can create computational templates that describe in a form that is understandable both by people and by computers what are the ways in which we want to describe the metadata to annotate biomedical data sets. We take those templates, and in the middle panel, we have a facility whereby scientists can describe their data by filling in those templates, by looking at the appropriate data types and automatically filling in the blanks with the right kinds of data, selecting terms from ontologies to make all the data consistent and standardized. And ultimately, as you see on the very right of the slide, we can take the acquired metadata, enter it through the template, and upload them along with the corresponding experimental data into a public repository where the data are stored and archived and where the metadata are now consistent with some community-based standard and filled in with standardized values, which makes the metadata findable, accessible, interoperable, and reusable. You'll notice in this slide we have a third panel where we are able to create a local cache of the metadata that we're uploading to those repositories. And we use this local cache during the time when we're filling in data sets to create new metadata. That cache allows us to learn patterns in previous metadata, and we can use those patterns to make suggestions to users as they create new metadata, basically facilitating the metadata entry process and making it a lot easier and a lot faster. So here is the library of metadata templates that Cedar offers, and this is just one user's particular library. Cedar allows users to have their own library, to share their templates with other users, to take public templates from other groups or users and use those as well. And if we wanted to, for example, click on biosample human, a description of these metadata or samples that are going to be submitted to the biosample repository, they're going to be human, and we click on populate, we can actually see an existing template that's been entered through Cedar. And what we see are the kinds of attribute value pairs that I showed you earlier. Sample name is 56, organism is Homo sapiens. And all of these are entered through the Cedar workbench during the step when we are filling in that template. So the question becomes, how do you create such a template in the first place? The left column in that diagram shows the step of entering the template information. And what we do is we have a whole environment whereby users can say, well, if we're entering biosample human template data, then there's going to be a sample name, which is going to be alphanumeric. There's going to be an organism, which is alphanumeric. There's going to be a tissue, which is alphanumeric. And notice by clicking on that little triangle beneath saying values, we're saying that those values are going to come from an ontology. And we can automatically find out what are the ontologies in that bioportal ontology repository that I showed you earlier that have tissue as one of the types of elements. And what we can see if we go to Bioportal, this diagram shows us, or rather this image shows us, that Uberon is an ontology that deals with tissues. We can see that MA, which is the mouse anatomy ontology, deals with tissues, but we wouldn't want to use mouse anatomy if we're talking about human anatomy. NIF standard, which is a neuroscience ontology, deals with tissues, and so on. Let's assume that Uberon is where we want to go. What Cedar allows us to do is to say, okay, if we want to use Uberon, let's make sure that's really the ontology we want. Cedar is able to download a portion of Uberon from the Bioportal repository. We can look at the terms that are in Uberon and confirm, yeah, that looks like it's a pretty good ontology for us to use in this situation. And by clicking on Add, we'll be saying that Uberon's branch dealing with tissue is going to be the set of standardized terms that we want to use for the particular metadata template that we're creating in Cedar right now. So we can create a template in the middle panel. We can fill in that template. And for example, here's biosample human again. And notice, for example, if we want to fill in the tissue, we said that the elements from tissue are going to come from the Uberon anatomy ontology and the portion of that ontology that deals with tissues. And we see the possible elements in the drop-down menu: blood, liver, bone marrow, breast, lymph node, and so on. What's cool is that the order in which those elements are listed represent the frequency with which they're going to be selected in previous metadata entries that Cedar has processed. And we see that in about half of the previous metadata entries with the elements that we already have filled in our template, blood was the choice that people were making, and to a much lesser degree, people might select liver or bone marrow or breast. And so by ordering the elements in the drop-down list based on the patterns that we see in the old metadata, we make the job of filling in the new metadata a lot simpler. What that means is, for example, if we're still filling in biosample human, but we're dealing with a tissue that is lung, and we're filling in the value of the disease, well, the disease in this case comes from DOID, which is the disease ontology. And we see when the tissue is lung, then lung cancer by far and away is the most common disease associated with that particular tissue specimen. And chronic obstructive pulmonary disease and squamous cell carcinoma of the lung are also up there, but lung cancer is first. And again, because we understand the previous metadata and can learn from them, we can make it really easy in our user interface to make selections that are most appropriate. On the next slide, you'll see what happens if we select or we indicate that the tissue is not lung, but brain. And if the tissue is brain, then the drop-down list includes things like Parkinson's disease and CNS lymphoma and autistic disorder. And frankly, the kinds of things you'd expect to be diseases associated with tissue that comes from the brain. And so again, we can use our knowledge of previous metadata and learning patterns from those metadata to make the job of filling in these templates easy. And even though the templates are more demanding than filling in an Excel spreadsheet, they make it easy to make sure that the data are correct, that the data come from standardized ontologies, that what's supposed to be a number is a number, that what's supposed to be a special symbol is a special symbol, and it makes it such that our metadata are better, making our data sets more fair. So Cedar really is a metadata-centric architecture. Our goal in Cedar, of course, is to make metadata and data more accessible online. But the thing to emphasize is that everything you just saw in that PowerPoint demo, all the semantic components, the templates, the ontologies, the value sets, all of those are managed as first-class entities, either in Cedar or Bioportal. And the user interfaces that you saw in Cedar are not hard-coded, but rather they're generated on the fly based on Cedar semantic content. So anytime the user says, I'm going to be filling in metadata based on a particular template, we automatically generate the UI for acquiring those metadata. And we can do that in a way which makes it really easy to fill in the blanks and create a metadata description that we know is going to be compliant with the reporting guideline that is represented in the minimal information checklist, if you will, and also is going to be compliant with all the ontologies that the authors have said need to be included in metadata to make them accessible. All the software components in Cedar have well-defined APIs, and people access Cedar not only as an amalgam, but also for the individual components. And what's great for us is that Cedar is able to generate all the metadata in JSON LD, giving us the opportunity to use the kinds of semantic technology with which you're all familiar to be able to access and process the metadata, to access data sets, to integrate data sets, to make sure that data sets are correct. So what does all this mean? It means that we no longer need to have spreadsheets as the basis for entering information, making those spreadsheets go away and basically allow users to be able to enter information using the kinds of structured, semantically rich elements that are acquired through the templates that Cedar manages. We are in a situation where we are no longer at the mercy of scientists who enter all kinds of ad hoc values for their metadata. Those ad hoc values go away because Cedar makes it trivial to make selections from the appropriate ontologies, allowing the terms in our metadata to be structured, to be correct, and to be searchable. So we know that although there's all this hype about making data fair, and although there's enormous desire to be able to ensure that the data that scientists put online can be findable, accessible, interoperable, and reusable, that's never going to happen until we can standardize the metadata that are used to describe the data sets. And we can't standardize those metadata until we're all using common templates that make sure that when an investigator tries to find a data set, he or she knows what to look for. And it won't happen until those templates get filled in with controlled terms from standard ontologies. So again, we know what to search for to access the metadata that we're interested in. What's interesting is that we know that with Cedar, we can create technology that makes it really easy for investigators to describe their data sets in standardized, searchable ways. And frankly, we think this kind of an approach is increasingly going to make it possible for the scientific enterprise to adopt semantic technology that's going to make it easy and direct to get access to the kinds of data that will allow us to verify experiments and to ensure that we can take other people's data and learn from them in ways which is simply not possible right now. So where is all this heading? Well, we know that once we can take the metadata that are needed to describe data sets and author them using this kind of data-centric technology, we can view the creation of metadata to describe data sets as a kind of publication. And ultimately, I think it's going to be that we're going to see the publication of metadata in a very rich form that intelligent agents can understand, being even more important, perhaps, than the publication of information in prose form in the guise of journal articles. And ultimately, if we can have semantically rich metadata that are standardized and predictable, then intelligent agents are going to start searching the web and, if you will, read the literature by examining the metadata that accompany online data sets. And those agents will be able to integrate the information in ways that is simply not possible now. They'll be able to track scientific advances, tell us when new experiments have been done that we want to know about, help us to re-explore existing data sets, and suggest a new set of experiments that might be the next logical consequence of the experiments that other investigators have already done. And if you believe what's happening now in scientific robotics, maybe we'll not only be able to suggest the next set of experiments by reading the data offline, maybe we'll even be able to do them robotically. And so that's sort of an overview of Cedar, technology that we're deploying in various aspects of science, all designed to make data findable, accessible, interoperable, and reusable, making the metadata that describe those data sets more available to the user community, and at the same time doing this all in a way which is remarkably data-centric.